Hi everyone. I hope you had a fun discussion yesterday, and I hope everything went well. I think your GSIs are doing a pretty awesome job. Please keep in mind, we're kind of figuring stuff out as we go. We always are trying to do our best to make sure you guys have a good experience. And if you have any problems or anything you're concerned about, definitely always let us know. So what I think I'm going to do with regards to the slides and the lectures is to split them up into two. Okay, and the reason is because if I have too many slides, it becomes really cumbersome and the files kind of have a fit when I try to upload them to YouTube. So don't be surprised when lectures for the day, there should be, most likely will be two of them per day. Okay, and the first hour and then the second hour. But I'm hoping everything goes okay. There is a video in the next slide. I had problems uploading videos yesterday, so if it doesn't instantly play for you, I will make sure you guys have the link to it. And I think that's it for now. So if you have any questions or problems, let me know. Otherwise, I guess we'll go ahead and get started. So the purpose of this lecture and what we're going to do today is to teach you about science, okay, in general. And so we're going to deal with hypotheses, and we're going to deal with experimental designs, and hopefully you guys just watched a video on buttered toast, okay? So Mythbusters, I will say I missed that show. I did like it. What I want you to be able to address, okay, stop the lecture right now and make sure you can address all of these questions, okay? What was the hypothesis they wanted to test? A lot of times, hypotheses, by the way, just so you guys know, I'm old, as my daughter likes to remind me. And so I was trained to use hypotheses in if-then formats. Okay, so what you might consider is if the toast is buttered, then I would expect to see da-da-da-da, or some hypothesis along those lines. Okay, so that is a hypothesis, generally if-then. I've also been told that it doesn't necessarily have to be that way these days, okay, in science. And so you can have hypotheses that don't necessarily appear as if-then. My response to that, by the way, when people tell me I'm old, I'm too old-fashioned, is that I had to use that method to fund my own research as a graduate student, where I used the if-then phrases to fund my research that cost about $250,000. So my response when I'm told that it's too old-fashioned is, it worked for me. Okay, but just realize it doesn't necessarily have to be done that way. Now, an experimental design means how many times did they drop the toast? How many times did they put an X on the toast? How many times did they butter the toast? How far, what was the height that they buttered, that they dropped the toast from? All of that goes into your experimental design, okay? Did they have a control? Yes, they did, by the way, okay? If so, what was it? Well, the control was the toast that was not buttered. All right, what were their results? So basically, what did they find? Of the number of times they dropped the toast, how many times did it end face up or face down? Okay, and this is something I want you guys to go through. And if you need to rewatch the video, go rewatch the video. That's fine. Okay, then you should also be able to determine what were their conclusions. Okay, and then, so what did they find? What did they conclude? Now, the last part of this is, were their conclusions supported by their results? Now, this might seem silly. Okay, but a lot of times in science, sometimes researchers, if you're trained well, you don't do this, but sometimes people can over-extrapolate on their data. Okay, in which case their conclusions may not be supported by their results, and that's when science gets sketchy. Hopefully, people who are trained well, that doesn't happen. Okay, but just realize every once in a while something slips through, and it may not happen completely. Okay, but again, make sure you can address all of these questions. Hint, hint. All right, and so then I think that will be it for this particular slide. So the whole purpose of science, by the way, is to basically learn about what's going on around us. So we're trying to explain the natural world. Now, we do this by using evidence, okay? And so, we test hypotheses. If it doesn't pass, we reject the hypothesis, all sorts of other good stuff. And that's part of, this scientific process that, by the way, took hundreds and hundreds of years to develop. Something else that is incredibly important with regards to science is that you have to be peer reviewed, okay? And if you're going to test a hypothesis, one of the most important components is that your hypothesis has to be falsifiable, okay? You have to be able to either find support for your hypothesis or find support against it. If your hypothesis is not falsifiable, it's not a good one. Write that down, okay? And by the way, I've mentioned this before. When I tell you to write something down, write it down. Now, in order to figure out how nature works, okay, and figure out what's going on around us, we ask questions. That's the first step. The first step to the scientific method is to ask questions. You should write that down too, okay? The second step is to propose a hypothesis. Now, hypothesis is generally a potential explanation. And once again, what does a hypothesis have to be? You should be able to tell me. Okay? Then you design an experiment to test your hypothesis. You conduct the experiment. You collect your data, analyze your data, reach your conclusions. These are all different parts of what it means to conduct science and the scientific method. All right? So write this down. The scientific method has several steps. First off, you ask a question. Secondly, you propose a hypothesis. Third, you design an experiment. Fourth, you conduct the experiment. And fifth, you collect your data. Sixth, you analyze your data using statistics. Okay. And then 7, you try to reach your conclusions. And the cool part about science, by the way, is very often when we find answers, there's a whole bunch more questions that we have, which is actually what makes it pretty cool. All right. Now, there are many different types of experiments. You can do an observational study where you're just looking to see what's going on around the world. Or you can do an experiment where you're actually manipulating variables. Okay, so a true experiment where you're experimenting with things, you tend to manipulate variables. It means you're changing conditions. Now, more recently, and when I mean recently in my lifetime, computers I'm sure have been around for most all of your lifetime, people have started to use computer simulations and mathematical models. And those can be incredibly useful because they allow you to make inferences over things that are a much longer term than we could even potentially exist, okay? So you could use a computer model to project a forest, 200 years from now after we're all long dead. All right, that's why those things tend to be beneficial. So just realize that, to test and evaluate hypotheses, you can do all 3 of these things, okay, that you use evidence from them. Okay, so a hypothesis, by the way, is a testable, possible explanation for a natural phenomenon. Now, it's a tentative answer to a question. And oh, look, a hypothesis must be falsifiable. So it has to make predictions that can be critically tested by experiments or by observational studies. And ignore the Launchpad thing there, by the way. We don't use Launchpad. Not for this particular section or for this particular class. If you guys take 172, they're going to make you purchase and use Launchpad. But I'm cheap, and I'd rather you guys have the cheaper version as well. And so I tend to think we'd get enough out of lecture that it's fine. So with your hypothesis, you might potentially confirm a prediction, okay, that supports that hypothesis, or you might negate it, okay, meaning that particular hypothesis was wrong. And that's all part of science, okay, and it's all information, and it all helps us reveal what's going on in the natural world. All right, so as we test ideas that are part of the process of science, we can also come up with what are called predictions. Now, predictions are relatively new in science, I gotta say. Not really all that impressed by them. But basically what you're doing is you're describing an expected outcome of your test, okay? And so you're predicting what's going to happen. Now, again, you can do science perfectly fine without predictions, but what I'm trying to do is train you guys in the most up-to-date methods, in which case, despite the fact I'm old, people use predictions these days, and so I want you guys to be trained in predictions as well. So the prediction, you're basically going to predict what you will see, okay? And so it'll make a little more sense as we keep talking about this throughout the lecture today. Okay, so let's actually apply this information. So let's say we have a research question. In what habitats are white-footed mice most common? And you put out 30 humane rodent traps, which means that they're live traps, so you don't hurt the little guys, okay? You put them in the 3 different habitats that you're testing. Okay, so you monitor the traps every day. Of course, you have to because you don't want anybody to die. You calculate the average number of mice in each habitat, and then you compare those averages with a statistical test. All right, so let's say this is your experiment that you're running. And so scientists is what we do, okay? Now, is this research best described as an observational study, an experimental study, a simulation study, or more than one of these? Now, this is not a type of test question you're going to see as far as like multiple choice. But what this is meant to do is have you apply things, okay? So if you choose to take 172 and everything goes back to normal in fall, where we're not online anymore, you might see multiple choice, in which case let's figure out how to address this, okay? So Is this research best described as blah, 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 blah? Okay, so take a moment, stop the lecture, read through the choices, and then I want you to choose one. Stop right here, okay, and then make your decision. Okay, so now we're started up again. All right, and so remember with an experiment, okay, well, actually, let me step back. A simulation study. This is not a computer simulation. Cross out C right away. Okay. Now, in an experimental study, you have to manipulate something. And traps, by the way, are not manipulating. All you're doing is taking a census. Okay. So you're not technically manipulating. So in addition to crossing out C, you can cross out B. Also, because of that, you can cross out D, which leaves us with A. Okay, so it is an observational study, and hopefully this makes sense because once again, for an experiment, you have to manipulate something, and trapping is not manipulating. All you're doing is taking a census of who's there. Okay, so let's say we conducted this experiment, and these are our results. Now, what's the first thing you do when you see a graph? By the way, the answer is not panic. Don't panic, okay? Part of what I want you guys to be able to do in this class is to learn to read scientific evidence and information. A lot of times we use graphs to show what we find. So the first thing you do, by the way, when you see a graph like this is you read your axes. What's the y-axis? That's the number of white-footed mice. What's the x-axis? That's the 3 different habitats, the forest, the grassland, and the suburbs, okay? Then you look at the levels. Okay, so look at the forest. How many mice were found? Look at the grassland. How many mice were found? Look at the shrubs. How many mice were found? Given that information, okay, you should feel pretty confident that you know what these results suggest. Okay, so summarizing the data from all the traps is best done in a graph like this. Now, the problem, by the way, of placing only one trap in each habitat, let's think about that. You're a scientist. You're conducting an experiment. One data point may not necessarily tell you anything. In fact, most of the time it doesn't. Part of science is that you have to have a large enough sample size. Write that down too, okay? In a good experiment, you have to have a decent enough sample size. So one trap is not going to cut it, all right? Now what I want you all to do is read through which conclusion is best supported by the data. Okay, so read through number A, least common in the forest. Well, no, doesn't seem to be that way. All right, B, least common in the grassland. Ah, that looks good. Okay, read through C anyway, just because. You want to make sure there's nothing there you're missing. White-footed mice, least common in suburbs. What's the correct answer? B. Okay, hopefully that makes sense. And again, all you have to do is read the graph and just interpret it as it is. Piece of cake. So let's talk about correlation and causation. Just because you have a couple of conditions that seem to be correlated, that doesn't mean that one is causing the other, okay? Now, one of the most controversial correlation and causation cases that's been around lately is the presence of autism, all right, and the amount of mercury in the vaccines, okay? So let's talk about this. It's really important as we go along in class that you not just learn the information, but you learn some of the history behind science, especially when things are done incredibly wrong, okay? Now, the whole idea about vaccines causing autism. So let's talk about how that came about. There was a man by the name of Andrew Wakefield, okay? You might have heard of this man. He is not a nice man. He's not well respected. And I've been in science now longer than you all have been alive. I hate saying that, but it's true, okay? He is one of the very few people that I have heard about getting kicked out and is no longer allowed to practice science. I have never heard of this before him. What does this tell you? He did something horribly wrong, okay? So the story behind all of this with the vaccines and the cases of autism. Autism it's a very frustrating disease for everybody who has it, okay? It's just, it's tough. And people want to know what's actually causing it, okay? Because that way, children who have it sometimes have a hard time maneuvering and functioning in society. And of course, we all love the children, and we want them to be happy and be able to do well in the world and so forth like that. And so, autism is something that we don't want to be around. So finding the cause of it would be great, okay? Because that way we can eliminate it. By the way, there are actually some recent studies that suggest it might even be the microbes in your gut, okay, that make a difference with autism. But again, more research has to be done, okay? So realize that this is a very controversial issue. A lot of research still needs to be done, and there is no easy answer to this, all right? Now, the story I want you all to know behind this is with Mr. Wakefield. So, Mr. Wakefield, apparently, according to some very well-respected doctors, was in cahoots with a pharmaceutical company. Okay, and he came up with this hypothesis that vaccinations cause autism. All right. Now, here's the problem. First off, his graduate students ratted him out as not sampling properly. You might ask, why would they do that? Well, when you have a graduate student advisor, your work tends to be correlated and connected to them, okay? You want to make sure that they're practicing good science, all right? And so if you end up someday going to grad school and your PI is performing sketchy science, you should run like the wind, I'm telling you now, because your work will ultimately get associated with them. So Wakefield was not sampling properly. He was taking samples from from children that were attending his child's birthday parties. Guys, that's not random, okay? That's not good sampling, and it's certainly not enough sampling. So he comes up with this theory, all right, that MMR vaccines, measles, mumps, and rubella, causes autism. The problem is nobody could replicate his results. Okay, so by the way, replication, having somebody else being able to replicate your results is crucial in science. And that's in order to have your data be trustworthy, that has to happen. Okay, so first off, his sampling was wrong. Secondly, nobody could replicate his results. These are two huge red flags. Third, his graduate students didn't even trust him. Okay, also bad. So a few doctors and scientific researchers started to do a little bit more digging. And what they found was Dr. Wakefield, I'm not sure how to refer to him, ended up being in cahoots with a large pharmaceutical company. And what they were doing was designing separate measles, separate mumps, and separate rubella, vaccines, that the whole idea is they would market this as not causing autism. Well, it's kind of the perfect scam because, of course, nobody who takes them would have autism because nobody who had the combined MMR vaccine would have autism either. Okay, so if you guys are curious about this, I can actually post some readings that suggest this was all for financial gain on his part. Okay, and the frustration is that he gives science a bad name. All right, not only that, children have died from diseases because he marketed this. That's why he is forbidden to do science anymore. Okay, so he's a perfect example of someone who was practicing for evil and should no longer be allowed to do so. When you start causing, so many children to die, anybody to die, knowing that you're running a scam, that's just unforgivable. I'm sorry. It just is. But, trying to keep my personal bias out of it. But you guys should look up the information online. Make sure that when you do, they're reliable sources. So anything.edu, anything.gov, I hope, is going to be considered reliable. Anything that is peer-reviewed and written by the experts. And after I'm done with lecture here, I'm going to try to find the sources that I found that were published, by the way, by doctors in reputable journals, that kind of revealed what he was doing. So long story short, I guess it's a very long story. Just because something is correlated doesn't mean that one thing causes the other. Another great example, by the way, where correlation does not necessarily mean causation. Here we are looking at divorce rates through time, and we're looking at the percentage of people consuming margarine. You can imagine there's this weird correlation. Nobody really knows how to explain it, why there might be a divorce rate in Maine that seems to vary with the consumption of margarine. This makes no sense, all right? It just happens to exist. A really good experimental design, by the way, can distinguish between correlation and causation, but you can imagine it's really hard to do with humans because you can't really experiment on humans. No one's going to do that. We've got a bad history of that. It's a bad idea. Don't even get me started on history of that. Okay, so you cannot cross those ethical boundaries for any reason. Okay, and so you have to conduct your experiments carefully and basically just observe what's already going on. And with humans, that's pretty tough. Okay, so margarine divorce rate, I don't know why this exists. It makes no sense whatsoever. Guarantee you it's not, just because they're correlated doesn't mean one's causing the other one. All right, so let's talk about a well-controlled experiment, okay? So in a well-controlled experiment, you're only going to alter one single variable in the test group. So it might seem simplistic, but that's how you get your best results. And a lot of times the most elegant experiments are those that are the most, very straightforward designs. And I love those, okay? You also then have a negative control where the variable is not changed. All right, so I want you guys to realize that in an experiment, very often we design them to have a negative control and a positive control. Okay, so with a positive control, you have a known effect and you're going to introduce it. Now, in a positive control, you want to see some change and you want to see your results change because you want to make sure equipment is working. Okay, or that your system is working. Now, in a negative control, you're not going to change that variable. And that's the whole goal. And ideally, you're not going to witness a change. Okay, so positive control and negative control. Now, not every single experiment that we ever conduct has a positive control and negative control. I used to work in the area of ancient DNA for my research. I can tell you we ran more negative controls to try and monitor for contamination than we actually ran for samples. But we also ran a positive control to make sure that if our reaction worked, okay, or to make sure if you do the positive control to make sure that your equipment is actually working. All right, so again, we're going to apply this more, and this will make sense more as time goes on. So just for right now, just realize that in a positive control, things should work. Your known effect should happen. In a negative control, you're not changing your variable, so things should not really change much. Okay, so let's apply this information. We're going to talk about a simple experimental study. Okay, our research question: is composted seaweed an effective fertilizer for tomato plants? That's the question. So let's think of a hypothesis. If composted seaweed is an effective fertilizer for tomato plants, then what would you expect to see? Okay, we'll come back to that in a moment. So in group 1 of our experiment, you'll have tomatoes planted in good potting soil and watered. You're going to have 20 of them. N is 20. That's your sample size. You should know this. Okay. Group 2, the same as group 1 plus Miracle-Gro. By the way, Miracle-Gro is awesome. Speaking as a gardener, they really have that stuff figured out. Okay. You're also going to have 20 separate plants. That's your sample size of group 2. Okay. Now, group 3 is the same as group 1 plus composted seaweed, N equals 20 once again. So let's ask this question. First off, what is your total sample size for your experiment of all 3 groups? You should know this. 60, okay, because you have 60 pots altogether. Now, you're going to grow your plants for 90 days, and then you're going to measure plant size at the end. So let's go back to your hypothesis. If composted seaweed is an effective fertilizer for tomato plants, then I would expect to see what? Either the plant height should be the same as Miracle-Gro or taller than Miracle-Gro. Okay, that's what your hypothesis would be, and that's what we're testing. Hopefully that makes sense. If not, let me know. All right, so as we're chugging along, let's keep applying. Okay, which group is the test or known as the experimental group? Group 1? Group 2 or group 3? What do you guys think? It's group 3. That's what we manipulated with the seaweed, and I hope that makes sense. If not, definitely let me know. So that's our experimental group. Okay, write that down. Now, let's think about this and go back to think about our controls. Which one is our negative control? In the negative control, you don't add the variable, remember? So which one is the negative control? Give you a hint, it's group 1. Okay, in group 1, you just have soil. Now, the other thing you should be able to answer, by the way, is what is group 2? Okay, so make sure that you then know what group 2 is. By the way, group 2 is your positive control. Okay, so your positive control, you're adding Miracle-Gro, you're going to get an expected result that those plants are going to be taller than your negative control. So Group 1, negative control. Group 2, positive control. And Group 3 is your experimental test group. All right. Now, the other two concepts you guys should be able to know is what an independent variable is and what the dependent variable is. Okay. Now, the way I want you to remember this is the way I was taught. The dependent variable depends on the independent variable. Okay. So the independent variable is what you're going to manipulate. In this case, what did we manipulate? We manipulated fertilizer. All right. That is the independent variable. The dependent variable depends on that. So what were we actually measuring? The dependent is what you measure. In this case, plant height. Okay? Make sure you understand that. You're going to see this again. Write this down. Did I mention to write this down? Okay? So the dependent variable depends on the, the dependent variable depends on the independent variable. The independent variable is what you manipulate. The dependent variable is what you're measuring. Okay, so the independent is fertilizer, dependent is plant height. Hopefully that makes sense. If not, let me know. Okay, so I know a lot of you want to go to medical school, in which case let's apply this to show that biology, intro bio, does even apply to medical school stuff. Okay, so you may or may not have heard what's called C. diff. Okay, Clostridium difficile. It's a bacteria that's often found in human intestines, and sometimes if all the rest of the bacteria in the gut is wiped out, C. diff will take over, in which case it's horrible. Okay, so imagine that you have to go to the bathroom 30, 40 times a day. All right, and it's not pleasant. All of the liquid gets pulled out of you. It's in the intestine. Nothing good comes from this. Causes a lot of pain, and if it's left untreated, it eventually causes death. Okay. Now, not too long ago, there were a group of scientists that found that, and you guys have probably heard this, the bacteria in our gut, of course, make a huge difference in so many different parts of our lives, including our mood and how we digest our food and, all sorts of other good stuff. And so, you want a healthy gut bacteria. That's why they have all those commercials of Activia and, gut flora and all, that type of thing. So what researchers found is patients who had C. diff, and by the way, there's a horrible smell associated with it. It's just awful. Okay. But patients who had C. diff actually could get over this relatively quickly if, in fact, they were treated with if they had another healthy donor stool sample that would replace their gut bacteria. Okay, so it sounds gross, but if you have this disease, you want to get rid of it. Okay, and what they can do is they can take a capsule and they can kind of custom design gut bacteria for you. All right, that can help you get rid of the C. diff infection, and it's a very, very wonderful thing. And it's called a fecal transplant. Okay, so we'll talk about it in the next slide. Okay, so once again, C. diff, researchers have found, can potentially be cured with a fecal transplant. Now, don't get grossed out. This is science, and this is just the way it is. Okay, so, and if you happen to be curious, by the way, the publication is there, and you guys are welcome to go to the library and download it and read it for yourself. And in fact, I highly suggest it. So a research study was done, and there were 3 treatment groups. Okay, the first one is the treatment group was donor feces, vancomycin, which is, by the way, an antibiotic, and what's called a bowel lavage. Now, a bowel lavage means flushing the bowels. Okay, that was group 1. Now, in group 2, the vancomycin, which is once again the antibiotic, plus the bowel lavage, which again, flushing the bowels. Okay, and then group 3 was vancomycin only. All right, now remember, the way a lot of people get this is to have an antibiotic wipe out all of the good bacteria, in the gut, in which case then C. diff takes over, okay, and that's what causes people to get sick. And the only way to get rid of it, all right, is to kill off the C. diff, and then, according to what this particular study found, add donor feces. Don't get grossed out, it's medicine. Okay, so given this information, okay, researchers found someone to be cured if they had no more diarrhea associated with C. diff for 10 weeks. And by the way, people who suffer with this will suffer for a long time until they go to the doctor, and sometimes, as I said, it can be fatal. If you take the donor feces, you can be cured overnight, which is pretty amazing. Okay. Given this information, what is the prediction for group 2? So let's think about this. Vancomycin and bowel lavage. So again, the antibiotic and flushing of the bowels relative to group 1. Okay. Well, remember group 1 is the donor feces. Now, it's not meant to trick you. Read the question. What do you expect to see for group 2? Well, if it's the donor feces doing, the fixing, then A is out. Okay. For B, compared to group 1, lower rate of cure of Clostridium. That one actually sounds pretty good. Keep reading just in case. For C, compared to group 1, equal rates. Well, that means the donor feces wouldn't work. Okay. And D, compared to group 1, a higher rate of cure. Well, then why are they even doing the study with the donor feces? What does this tell you? If you ever see a question like this, you would go for answer B. Okay? If that doesn't make sense, let me know. All right, so let's keep applying. Okay? Which group is the positive control? Now, in the positive control, okay, you're not changing the variable, so you're not adding the donor feces. All right? What this means is that your positive control has to be group 3. All right, you're just doing the vancomycin or the antibiotic. You might be asking yourself, well, what's going on with group 2 and why did they do that? And I would say, aha, that is an excellent question. Go to the next slide. Okay, so why did they include group 2? Well, the reason they did is they had to make sure that if people were cured, it wasn't the bowel lavage doing it. Okay, so they had to have group 2 to double check to make sure that it was actually the donor feces that was the cure. All right. Now, if you added a 4th group who received no treatment, okay, what type of control would this be? All right, what do you guys think? The answer is B. Okay, so if you have patients who receive no treatment, that would be a negative control. Because remember, in a negative control, okay, you would expect that nothing's going to change because you're not manipulating your variable. All right. Hopefully this makes sense. Okay, and hopefully to apply it. Those of you that are gunning to be doctors, go read the paper. It's good for you to do that, especially now. Okay, so as we're discussing science, let's talk statistics. Now, I don't know about all of you, but when I was an undergrad, statistics was the bane of my existence. Okay, it has taken me so long to be able to learn, appreciate, and feel confident in statistics. Okay, and honestly, it didn't happen for me until grad school. So, you might be asking yourself, what's the purpose of statistics? And I would say, yes, I understand that question. The reason we use statistics is that we want to test our hypothesis and be unbiased, okay? And so a lot of times in science when we're conducting experiments, we really want to see a result, okay? And so if we're looking at the data, we might try and force it and say, oh, look, I think it's a positive result. I think it's actually working. And the reality is it might not be. It might just be something random, okay? So we use statistics to be honest and to keep ourselves honest as scientists and to be able to see, is our treatment really making a difference? Okay, knowing that information, the other thing I want you to know is that it's better to have a larger sample size if you are running an experiment. Now you have to realize that there are limitations, money, time, all that other good stuff, but the larger sample size you have, okay, the more you can truly test your hypothesis and feel confident in your result. The reason that happens is because your standard error will decrease, okay? Now, standard error is basically just like random error in an experiment. And so you get these wild data points that just happen for whatever reason. You want to minimize those, okay? So you take the average, and the larger your sample size is, the smaller your standard error decreases, and the more confident you can be in your result. Okay, that's why we use stats, and this is definitely a relationship I want you guys to know. Increase in sample size, decrease in standard error. All right, guys, so I have made a hobby out of learning how people study. And in fact, I've done this so much so that I designed a class on it, which we offer here at Michigan. The good news is that this is a hobby of mine. The bad news is I'm going to tell you what I've learned. Actually, it's good news for you, okay? Every once in a while, you might hear my husband pipe in. Realize I don't have an office anymore. If you hear dogs barking, I'm sorry. Okay. Or if my husband pipes in, still sorry. All right. So, the fun part about this, though, with regards to learning how people study, is that, I read up on the research. So, the question that I was curious about, what's the most effective way to learn new material and actually keep it? Okay, so they took 20 college students, I'm sorry, 4 groups, in each group 20 college students. Total sample size of what? You should know this. 20 times 4. Okay, then they were asked to read a passage from a textbook. They were asked to read it once, that's the study group. They were asked to read it 4 times or more, that's the second group. They were asked to concept map. Now, I'm not saying, by the way, that these things don't work, but the question was, what is the most effective way? And then the last part, the last treatment group, was called retrieval practice. This means that they read it once and they try to remember everything from memory. Okay, then they type it in a computer, or they take, my suggestion for this, by the way, is to pull out a blank piece of paper and write down everything you know about a concept. Okay, so be familiar with these 4 different ways. Study, repeated study, concept mapping, retrieval practice. So let's you think about it. What do you think is going to be the most effective? Studying for 25 minutes, repeated study. C, which is concept mapping, or D, which is retrieval practice. Okay. Now, before I learned about this, by the way, I would have said B or C. Okay. But let's actually talk about what has been found. Now, based on, what students have expected in the past to be most effective, and this was back when I could offer this class live, okay, this was the pattern. Students felt that repeated study would be the most effective in learning new material. And that's a reasonable hypothesis and reasonable, potential answer. But let's talk about the real answer. Now, speaking as a scientist, whenever I'm told something, the first thing I say is show me the data. Okay, in which case, when this study was actually conducted, all right, and people were asked to recite verbatim questions from the material, okay, that's graph A. So which method works best? The retrieval practice. Okay, now when it comes to inference questions, in other words, how did they interpret something and so forth, once again, retrieval practice works. Okay, so my suggestion to you all, when you're first learning something, read it the best you can. Try to interpret the best you can, but to make it an active reading, and we'll talk more about that, how to do that later in the semester. But then if you truly want to figure out how much of something you know, pull out that blank piece of paper and just write everything down that you know about it. Then whatever you're missing on that concept, that's what you need to study on. Pretty cool, isn't it? So, based on the results presented in the previous slide, which technique works best? D, retrieval practice. Okay? Just a little side, information and helpful suggestions, especially when you guys ramp up for your later on science classes. All right, so part of science and what makes science successful is the ability to publish your work, make it available for peer evaluation, so then other scientists can try your experiment and then hopefully replicate your results. Okay? I will tell you, as a scientist, you get pretty nervous when you put stuff out there because other people are going to test, your results and check to see if you're correct. And I will tell you from personal experience, I published my first chapter of my thesis when I was in grad school. I was pretty nervous, okay? You're putting yourself out there, but we did well and got it published. A second group came along, conducted a similar study using a different set of genes, different samples, different everything, found the same thing I did, which meant my results had been replicated, okay, and I did a happy dance all the way down the hallway. Okay, so when you're nervous and you're just publishing stuff out there, especially when you're a young scientist trying to learn, it's really nice, okay, to be able to have others replicate your results. Now, the other thing I want you guys to realize is that science itself as a field has evolved over hundreds of years, okay? It's not like these rules were just created all at once and it was like, okay, if you're a scientist, you have to do this, 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 and this, okay? We're dealing with hundreds of years of research and researchers before us who've tried to address questions and run their own experiments, and so this is where science sits right now, so it's had hundreds of years to be tested and so forth, so that's a really good thing, which is part of the reason that it's so cool. Okay, the other thing I want you guys to realize is our knowledge in general of biology has changed over time. Science is constantly self-correcting. We're always challenging what others have found. That's part of what's so cool about it. This, overall, over time, will change things that we know and understand about the natural world, and our understanding improves. That's why it's such a really awesome, cool, exciting field. And, not that I'm biased or anything, but I totally am. But that's okay. It means I like my job. All right, guys, time to apply this. Okay, you should be able to take a prediction, figure out which letter is a prediction. Theory, figure out which letter. And hypothesis, figure out which letter. Okay, now there are hints in each of those letters with those descriptions. Okay, put down your best answer. Make sure you feel confident about it. You're going to be fine. So we all know that scientists and sometimes the general public have a hard time communicating with one another. Okay, and there's various reasons for that. The reality is we sometimes use terms very differently. One example is theory. Okay, now I have a theory that it's going to rain tomorrow. If we're using this in colloquial, in everyday speech, I don't know. There's no evidence to back that up. I haven't checked the weather. That's a total guess. Who knows? Okay. But in science, a theory is backed up with proof. It is supported. It is, modified as need be. It is always open to falsification. Okay? By the way, you should never hear someone say, I have proven this. As soon as I hear that, I roll my eyes. Okay? You can say, I've found support for this hypothesis or against this hypothesis. Okay? But to prove something, that gets a little dicey. All right? But the reason that the general public and scientists tend to not necessarily agree on everything, part of it is the language we use and the terms and how they can differ. So what I want you to get from this is that, first off, theories are essential for science, but that if you're using a theory in everyday colloquial speech, you don't have to have support for it. If you're using it in science, you definitely have to have support for it. Write that down. Now, I'm bummed that you guys can't talk to your neighbors, okay? But given what we've discussed so far, you should be able to figure out, what makes a good experimental design. This is an excellent application question. Make sure you can go through and address this, okay? And fill this out.